Namaste students. So today in Biomolecules chapter we will study regarding to the structure of DNA. As we all know that there are mainly two types of nucleic acids, that is nothing but DNA as well as RNA, where in case of DNA it is nothing but deoxyribonucleic acid, which is one of the major genetic material in most of the living organisms. So now regarding to the structure of DNA, which was mainly done by Watson and Crick, that is why it is known to be the Watson-Crick model, and it is also called double helix model. Okay, it is also known as a double helix model because of presence of two helices. There is a presence of two helices, so there are two polynucleotide chains are involved here, and it is also known as a secondary structure. Why it is called secondary structure? Where in the case of secondary structure of proteins, it is having a helical structure. So this is also a helical structure. That is why it is known as a secondary structure. So the structure of DNA, it is double helix model, Watson-Crick model, as well as it is a secondary structure. So here it is mainly consisting of two polynucleotides. So there is a presence of two polynucleotides. There are two polynucleotides, and these two polynucleotides run anti-parallel to each other. So this is parallel and this is anti-parallel. They run anti-parallel to each other. Okay, so there are two polynucleotides as well as they run anti-parallel to each other, and their backbone is mainly made up of sugar phosphate sugar chain. So this is nothing but their backbone, and this is made up of sugar phosphate sugar chain. Completely they are made up of sugar phosphate sugar chain. So perpendicular to this sugar phosphate sugar chain, okay, perpendicular to this there is a presence of nitrogen bases. Perpendicular to the backbone there is a presence of nitrogen bases. As we all know that DNA is mainly consisting of 4 nitrogen bases, 2 purines such as adenine and guanine, they are nothing but the purines, as well as 2 pyrimidines such as cytosine and the thymine and cytosine, which are the 2 pyrimidines, and adenine and guanine are 2 purines. So now here in the case of structure of DNA, where the purines are always paired with the pyrimidines. Purines are always paired with the pyrimidines, where adenine always pairs with the thymine, cytosine always pairs with guanine. Okay, adenine always pairs with the thymine, cytosine. So here you can see this is a detailed structure. In the detailed structure you can see where there is a thymine, and here there should be adenine. Okay, adenine pairs with the thymine, guanine pairs with the cytosine. So purines, the amount of purines as well as pyrimidines are equal. Okay, so now here, so that is regarding to their presence. So here you can see the adenine and thymine, there are in between two lines. This represents the presence of two bonds. There are 2 hydrogen bonds between adenine as well as thymine, and there are 3 hydrogen bonds between cytosine as well as the guanine. Okay, 2 hydrogen bonds between adenine as well as thymine, 3 hydrogen bonds between the cytosine as well as the guanine. So here in the structure of DNA there are 2 polynucleotides. These 2 polynucleotides run anti-parallel to each other. So it is from 5' to 3', 5' to 3'. So here 3' to 5'. That is their. So here they always, adenine is always paired with the thymine, cytosine is always paired with the guanine. Adenine with thymine, cytosine is with the guanine. Adenine is having two hydrogen bonds. In between adenine and thymine there is a presence of two hydrogen bonds. In between cytosine and guanine there is a presence of three hydrogen bonds. Okay, so that was regarding to the bonds. So as I told, their backbone is made up of sugar phosphate sugar chain. Their backbone is made up of sugar phosphate sugar chain. So perpendicular to that there is a presence of nitrogen bases. Perpendicular to that there is a presence of nitrogen bases. So the strand turns around 36 degrees. So they are turning, this strand turns around 36 degree. Okay, strand turns around 36 degree, and this complete pitch. So this complete pitch is about 334 angstrom. Okay, so the complete pitch is about 34 angstrom. So the each pitch is consisting of around 10 pairs of nitrogen bases. Okay, so if it is a complete pitch which will consist of around 10 pairs of nitrogen bases. So the distance between successive bases is nothing but 3.4 angstrom. Complete pitch is 34 angstrom. So the distance between these two is nothing but 3.4 angstrom. So they are arranged like a ladder. Okay, so the entire pitch is like a ladder. So the steps are represented by these particular nitrogen bases. Steps are nothing but the nitrogen bases. Okay, so now the amount of purines are always is equal to amount of pyrimidines. Amount of purines are always is equal to amount of pyrimidines. So that is regarding to the structure of DNA. So in the structure of DNA, which is known as Watson and Crick model, they got Nobel Award for this, and it is also called double helix model or the secondary structure. So in the DNA structure, it is mainly consisting of two polynucleotide strands, where these two polynucleotide strands run anti-parallel to each other and forms a helix. Okay, so here their backbone is made up of sugar phosphate sugar chain. Backbone is made up of sugar phosphate sugar chain. Perpendicular to this there is a presence of nitrogenous bases. So regarding to the nitrogenous bases, purines are always paired with the pyrimidines. Purines are always paired with the pyrimidine. Adenine pairs with the thymine. Guanine pairs with the cytosine. Okay, so here there is a presence of two hydrogen bonds between adenine and thymine and three hydrogen bond between cytosine as well as the guanine. The strand turns around 36 degree. The entire strand turns around 36 degree, and the one pitch is about having a length of about 34 angstrom. So the one pitch, so it is appears like a twisted ladder or the steps. Okay, twisted ladder. So here what happens with the nitrogenous bases represents the steps. Okay, that mainly represents the steps. So here the distance of complete pitch is around 34 angstrom. So the successive pitch is about 3.4 angstrom. Okay, so this is regarding to the structure of DNA. Okay, so the entire structure of DNA. So there is also a Churgov's rule. According to the Churgov's rule, the adenine will be always paired with thymine. Cytosine is always paired with guanine. Okay, as well as the amount of purines is equal to pyrimidines. Okay, amount of purines is equal to pyrimidines. A plus T divided by C plus D is equal to 1, which is not required, which is not mentioned in your NCERT. Okay, so that is regarding to the structure of DNA. Next we will move on to the dynamic state of the body, that is concept of metabolism. Dynamic state of body, that is nothing but the concept of metabolism. So now regarding to the concept of metabolism, so here we have a term that is nothing but turnover. See, when we talk about the biomolecules, those are nothing but the chemicals which are present in the living organism. Okay, chemicals which are present in a living cell, that is known to be the biomolecule. Now, in our body, we consider our own body. In our body, what happens is there are a lot of chemicals which will be produced. Okay, so the lot of chemicals are produced and one chemical will be converted into other chemical and that will be converted into one more. For example, the amino acid. Okay, so the essential amino acid we take it through food, non-essential amino acid which are synthesized in our body, these may combine to form a protein. Okay, protein will be having certain other functions, so that is nothing but turnover. Turnover refers to the chemicals or the biomolecules which are constantly converted into other and that forms a one more thing. So that is nothing but turnover. For example, glucose will be converted into glycogen. Glycogen again broken down to form glucose. All those are nothing but the turnover. Okay. So that is here overall concept is nothing but the metabolism. Whatever the turnover concepts are here and that is overall altogether known to be the metabolism. Okay, so now regarding to the metabolism or the metabolic basis for living. Metabolic basics for the living. So here metabolism is nothing but the overall, as you know that it is the overall chemical reactions which is taking place in a living organism and that is known to be the metabolism. Okay, metabolism, the reactions we can also do it in the laboratory conditions outside the system. But still, naturally the metabolism what we have, which is mainly consisting of two main processes, that is anabolism or anabolic pathway as well as the catabolism, catabolic pathway. Okay, conversion of simple substances into complex substances, that is nothing but anabolism, making. Okay, breaking down of complex substances into the simple one. Okay, breaking down of complex substances into the simple one, that is nothing but the catabolism. Disaccharides are broken down into monosaccharides, that is nothing but the catabolism. Monosaccharides are joined to form a polysaccharides, that is nothing but the anabolism. Okay, so removal of amino group from the amino acids, removal of carboxyl group, removal of carbon dioxide, all these are considered to be the metabolism. For example, these processes, the process of metabolism, that may be a single step process. The process of metabolism may be a single step process or it is a multi-step process. Or the process of metabolism may be a linear procedure or it may be a cycle. It may be a linear pathway or it may be a cyclic pathway. What do you mean by a linear pathway? What do you mean by a cyclic pathway? Linear pathway, one is converted into other. Cyclic pathway, one is converted into other, that will be converted into one more thing, so that the first whatever the substance was there, that we gain it back. So that is a cyclic pathway. Krebs cycle is there. So there are various cycles, biological cycles are there. Okay, so that is these metabolic pathways are either linear or they are cyclic. They may be consisting of single step processes or they may be consisting of multi-step processes. For example, the glycolysis, glucose to lactic acid, especially takes place even in our muscles. Okay, that may be a 10-step process. Okay, so the entire process will be around 10 steps. So that is regarding to the metabolic basis for life. One of the energy currency or energy in the form of bond. The living organisms are are mainly consisting of energy currency, and that is nothing but ATP. ATP is nothing but adenosine triphosphate. Adenosine triphosphate. ATP. So ATP is one of the energy currency what we have. Okay, it is the energy currency. It is like a debit card. ATP is like a debit card. We should have energy, then only we can spend. It will never be a credit card. After spending, we cannot fulfill it. Okay, so it is very important. It is in the form of a bond. It is also known as a bond energy. Okay, so that is regarding to the glycolysis as well as that is regarding to the ATP. Now there is one more thing is that even in case of dynamic state. So in case of these where the biological system where these will be in a non-equilibrium condition. Okay, so that will be in a non-equilibrium condition that will never reaches to a equilibrium. So there is nothing known as a final result. Okay, so our system, see though we may stop working but our system will never stop working. When our system stops working, ultimately we have to stop. That is nothing but a death. So here what happens is it is in a non-equilibrium state and non-equilibrium steady state, continuously working one. Okay, living systems, they are in a non-equilibrium steady state. So that is very important in case of the living organisms. All these are the small concepts. Normally 1 mark questions can be asked, nothing else than that. Okay, such as a turnover, what do you mean by catabolism, what do you mean by anabolism. As we all know that there is very important substances which we have are nothing but the enzymes. Okay, so the enzymes are the proteins. They are the proteins which are produced in our body. They are mainly acting as a biocatalyst. So regarding to these, was what I told, it is a non-equilibrium process. So for those processes, these substances are very important. Okay, those substances are very important. So one more thing is that there are a lot of substances that has to be produced in certain limits. So as we all know that blood pressure, even it is having a normal range. Blood sugar 4.5 to 5 micrometer. So that is also having a certain range. So each and every substances which are produced in the living organisms will be having a certain range, normal range. If it is increased or if it is decreased, that will be having an adverse effect on the living system. Okay, so that will affect the steady state of the living system, working of the living system. Okay, so that is regarding to these concepts. Okay, so next we will move on to the last topic in this chapter, that is nothing but enzymes. Okay, so next we will study about the enzymes. So this is one of the very important topic under biomolecules. So what are these enzymes? Okay, what are these enzymes? So enzymes are nothing but proteins. All enzymes are proteins, but all proteins are not enzymes. Okay, enzymes are the proteins which are synthesized in our body, which will speed up the chemical reaction without itself undergoing any change. There is no change in the enzyme, but it will speed up the chemical reaction. They will work on substrate and convert substrate into a product. Those are nothing but enzymes. Enzymes are the chemical substances which are produced. They are the proteins synthesized by the body which will speed up the chemical reaction without itself undergoing any change, and that is known to be the enzymes. Some of the nucleic acids behave as enzymes and they are known to be the ribozymes. Some nucleic acids, they behave as enzymes and that is known as ribozymes. ribozymes are some of the enzymes which behave, some of the nucleic acids, and they behave as enzymes. As I told, where enzymes, they are mainly acting as a biocatalyst. They will speed up the chemical reaction, especially inside the organism. Okay, without these enzymes, lot of functions will never take place. Okay, so they will be speeding the chemical reactions. So when we look into the enzymes are consisting of a protein part. As I told, it is made up of a protein. It is a protein. So it is having a protein part, and that is known to be the apoenzyme. Enzymes are consisting of a protein part, and that is known as apoenzymes. And the non-protein part is called cofactor. Protein part is called apoenzyme. Non-protein part is called cofactor. Together known as the holoenzyme. Apoenzyme plus cofactor give rise to holoenzyme. Now, there is a site in the enzyme for the binding of substrates. Okay, so there is a site in the enzyme for the binding of substrate, and that is known to be the active site. Active site, it is the site or the crevices or the pocket which is found in the enzymes for the binding of substrate. So that is known to be the active site. So as I told, enzymes are consisting of these cofactors. Sometimes without the cofactor, enzymes cannot work. They cannot function. When we remove the cofactor from an enzyme, it becomes inactivate. As we all know that for the functioning of enzyme, there is a certain favorable temperature as well as a pH. Okay, but even in the hydrothermal vents or in the hot springs where the temperature is very much high, there is also certain enzymes which will work in those conditions. Okay, so that is regarding to that. Now we will look into these, what are these cofactors. So now regarding to the cofactors are nothing but non-protein part of the enzyme. So regarding to these cofactors, they are mainly consisting of three types of cofactor. One is a prosthetic group, coenzyme, and the metal ion activators. Prosthetic group, coenzyme, as well as the metal ion activators. So the first and foremost regarding to the prosthetic group. Regarding to the prosthetic group, they are actually a non-protein substance which will be always found associated with the apoenzyme. Non-protein, they will be always found associated with the apoenzyme. That is the prosthetic group, permanently attached to the enzyme. For example, peroxidase and catalase, which will do the reaction of breaking down of hydrogen peroxide into water. Okay, peroxidase and catalase, they are the enzymes. Normally enzymes will have a suffix ase. Okay, so they will do the function of breaking down of hydrogen peroxide into water, where they have a prosthetic group known as a heme. Heme is a prosthetic group which will be found attached to this. So that is nothing but a prosthetic group where the non-protein which will be permanently attached to the protein part of the enzyme. They are required by the enzyme for its function. Without this, there is no function, enzyme becomes inactive. Whereas the second one is nothing but the coenzyme. So regarding to the coenzyme, so these are temporarily associated with the enzyme. Whenever there is a reaction, they will be found attached to the enzyme. When there is no reaction, they will be detached. Okay, when there is a reaction, they will be found attached to these particular enzyme only for the reaction they are required. Other than that, they will not attach with these. For example, nicotinamide dinucleotide phosphate, NAD as well as the NADP are consisting of a coenzyme known as a vitamin niacin. Okay, vitamin niacin, which are mainly a coenzyme which are temporarily found associated with this particular enzyme. When there is no reaction, they are not attached to it. They are no more a part of our enzyme. So, last type is nothing but metal ion activators. Okay, so there are certain substances which will be acting as a activators. They will activate the reaction. Okay, they are mainly acting as a activators. They mainly activate the reaction, such as metal ions, such as zinc, chloride, magnesium. Examples include zinc is acting as an activator for the reaction in the enzyme carboxypeptidase. Or the salivary amylase is having an activator, that is chloride. In our mouth, salivary amylase is present. Okay. So, for the carbohydrate digestion, so where the chloride is mainly acting as a activators, they will activate the reaction. Okay. See, these enzymes will also work without the activators. But if the activators are present, reaction will be faster. Okay, so that is regarding to the metal ion activators. They will activate the reaction. So these are the three types of cofactors. There are mainly three types of cofactors, such as a prosthetic group as well as coenzyme and metal ion activators. Prosthetic group are the non-protein part. They are permanently attached to the protein part. For example, peroxidase as well as catalase, which will break down the hydrogen peroxide into water, having a prosthetic group known as a heme group. Coenzyme, they are temporarily associated with the enzymes such as NADP as well as NAD is having a vitamin component known as a niacin as a coenzyme. Metal ion activators, they are mainly activating the enzyme actions such as zinc as well as the chloride. So these are regarding to the cofactors. So this is about the enzymes, where the enzymes are the proteins which are synthesized in our body, which will speed up the chemical reaction without itself undergoing any change, where the enzyme will never undergo any change. Okay, some of the nucleic acids acts as enzymes, and that is known to be the ribozymes. They are the biocatalysts, where the enzymes are the biocatalysts, where enzymes are consisting of a protein part, that is a coenzyme, non-protein part is a cofactor. Even the enzymes are consisting of certain pockets or the spaces, that is known to be the active site. Active site refers to the site which is meant for the binding of substrate to the enzyme. So enzymes, each and every enzyme is known to be the specific. When we look into the properties of the enzymes, okay, when we look into the properties of the enzyme, each and every enzyme is specific. One enzyme, one substrate. Okay, one enzyme, one substrate. Salivary amylase only acts on polysaccharides. It will not act on proteins. So that is one enzyme, one substrate. Maltase will act on maltose. It will not act on lactose. That is, enzymes are specific. As well as enzymes are temperature sensitive, thermosensitive. They are sensitive to the this particular temperature, except some of the inorganic catalysts. So inorganic catalysts slightly variant from these. Okay, but exceptions are there. Normally enzymes, if the temperature is very high, they will be denatured. As you know that proteins will be denatured, same way enzymes will also be denatured at the high temperature. If the temperature is very low, they will be inactive. Okay, so the normal temperature between 25 to 45 degrees Celsius, they will work well. Okay, so next is about, they are pH sensitive. They are also pH sensitive under certain pH, especially neutral pH is a favorable pH for the enzymes. If the pH is acidic, they will not work. pH is alkaline, they will not work. Exceptions are there, because certain enzymes even work in our stomach where the pH is acidic, even work in the intestine where the pH is alkaline. Okay, and they will do a teamwork. Group of enzymes will do a teamwork. For example, there is a digestion process, carbohydrate digestion process. So first what happens in the mouth, enzymes will work. Next, in the intestine, enzymes will work. For performing one function, all the enzymes will work in a team. So that is nothing but a teamwork. So these are the properties of the enzymes. Okay. So in the last session, we will discuss the remaining parts of the enzymes. Thank you.